Hello everyone. I welcome you all once again to my series of lecture that is Understanding Pharmaceutical Science with Dr. Hariharan. So today we are going to discuss about the types of fermentation process. So I have divided this lecture into two parts. Most precisely I have discussed about the different types of fermentation process and later on some of the terms which is very important in the continuous fermentation process. So first we will understand what is meant by a fermentation. So if you see in my previous lecture, I have told that fermentation is a Latin term called as fervor, which is look like a boil-like condition. So why this term has been coined? Because during the production of ethanol, what happens is a boiling-like conditions will be viewed because of the evolution of the carbon dioxide during the anaerobic condition. So, but this fermentation, if you see, actually it is a metabolic or biochemical process in which a microorganism utilizes certain substrate from its media, and this substrate is processed by the microorganism through its biochemical or metabolic pathway and produces a product. So what we are actually doing in the fermentation is we are providing a particular substrate to the microorganism and we are collecting the desired product. So this process we call as fermentation. So the products generally what we collect from this process is that either a primary metabolite or a secondary metabolite, which also having a industrial importance ones. So if you see what is meant by a primary metabolite, generally the primary metabolites are the metabolic products which is necessary and essential for the growth of the microorganism, like an amino acid. Whereas the secondary metabolite, which is not necessary for the growth of the microorganism, but it can support the microorganism, like fungi, the Penicillium notatum producing an antibiotic penicillin. So this penicillin released around its surroundings, what happened? It inhibits the growth of the bacteria, thereby the fungi will have enough nutrients for its growth. So this is how we will find this primary and secondary metabolite. So based upon the nature, we will collect from the microorganism through this process, that is the fermentation process. So we can classify the fermentation process. If you see the different types of fermentation process, we can classify basically into three types. One is the batch process, second is the fed batch process, and third is the continuous culture process. So first, if you see a batch process, the batch process, the term itself tells it is working as a batch. So batch means a specific number, specific quantity we can call it. So what happened in this fermentation process, we take a vessel which is a closed one. So in this closed system, what happens? We will add a specific quantity of a sterile media, then we will add the microorganism. Then it is kept just like that. So after a particular time period, what happens is that this microorganism, that is we called as an inoculum, which is added to this media, will grow, then it forms the product, and finally the product is recovered out. So this process we can better explain by the growth kinetics of this microorganism. So if you see the microbial growth kinetics, how it happens after adding a fresh inoculum into the fresh media in this batch process, it goes through a specific stages. So first, after a short particular time, the microbial cell will follow a phase called as lag phase. Then it started growing, it goes to a log phase, then it will be maintained at a specific quantity called a stationary phase, and if it is further allowed, then it can lead to the decline phase. But for our fermentation process, we will more concentrate about these three phases: lag phase, log phase, and stationary phase. So how these growth kinetics is very important in the fermentation, we can understand that depending upon our product, at which stage we have to get the product and which stage we can maintain to get the product in large quantities is depend upon this growth kinetics. So what happening actually is that we have taken a fermenter containing a sterile media, we added a specific volume of the inoculum, that is the microbial organism. So after adding this microbial organism, there will be no immediate growth take place because the microorganism has grown in a specific environment while the inoculum development. when it is added to a fresh production media which is having a different set of cultural condition, what happens? It needs certain time to adapt it. So this time we call as a lag phase. So in the lag phase, what happens? There is no growth occurs. What actually the microbial cell will do is it will adapt itself for growing. That means it produces necessary enzymes or its machineries, metabolic machineries, so that it can consume that particular substrate. So for a commercial purpose, we doesn't need a prolonged lag phase, because the prolonged lag phase will finally affect the production cycle. So the production time should be reduced. So we have to avoid this lag phase. So how we can avoid this lag phase? If we see that, we have to use a suitable medium which can reduce this lag phase. So for example, if I use the inoculum development, the same production media, it is used for the production media also, then there won't be any problem. So the bacteria, if it is added directly into the production media, it will give the same conditions. So there will be less lag phase, or even we can avoid the lag phase itself. So after the lag phase, what happens is that the rate of growth of the microbial cells will start getting increasing. So the cells will double after a particular time intervals, generally called as doubling time. So this phase we call as a log phase. So it produces an exponential growth. So it is also called as exponential phase. So if you see what happening in this phase is the microbial cell consumes the nutrients and the biomass will increase. That is, the population volume of the microbial cell will increase, and because of this it also fosters producing the primary metabolites. So if you see, most of the primary metabolites can be produced during this stage, that is the exponential phase. And after a particular time in this batch culture, what happens is once the nutrients has been utilized, the growth rate started decreasing and it reaches to a stationary level. We call it as a stationary phase. So technically, if we see, the stationary phase can occur by two ways. One, it can be due to the depletion of the necessary nutrients. The nutrients which is needed for the growth has been depleted. So this condition of stationary phase we call as substrate limitation. And the second way is that once it has consumed the nutrients during the exponential phase, that is during the growth phase, it also metabolizes it by the metabolic waste will come outside into the media, which can be toxic in nature. So this is called as a toxin limitation. So in the stationary phase, whether it can follow a substrate limitation or toxic limitation, we have to analyze it. So if it is a substrate limitation, then we can use other types of fermentation process to get the maximum yield. If it is a toxin limitation, then we cannot get a new approach so that the production can be maintained. So in the stationary phase, if you see, it will be a metabolically active, the bacterial or microbial cells will be, and it produces a large quantity of secondary metabolites. So the secondary metabolites such as antibiotics or solvents such as ethanol can be high production, can happen only in the stationary phase. So this picture, if you see that, so what actually the process is happening, initially the nutrient level glucose will be high, the biomass, that is the microbial cell, will be low. So as the time goes, it consumes these nutrients, then the curve will start elongating. So once it reaches the stationary phase, the product formation will start growing. So you can see both the curve. So if you see the application of this batch fermentation, it is used to produce either a biomass, primary metabolite and a secondary metabolite. So if you see, biomass is nothing but we are providing this cultural condition in such a manner that it produces a fastest growth and the maximum cell population. So in single cell protein manufacturing, we need a large biomass. Sometimes the biomass is very important for the production of the primary and secondary metabolites. Second is the metabolites, which may be a primary or secondary. The primary metabolites, I have already told you that the primary metabolites can be more produced in a huge quantity only in the exponential phase, because I have told you that primary metabolites is essential for the growth of the microbial cell. So if a microbial cell is growing, that means primary metabolite is producing in a large quantity. So in this stage it can be collected. So the secondary metabolite doesn't support the growth, but it can maintain the necessary supply of nutrients by excreting this secondary metabolite. So generally at the end of exponential phase and in the stationary phase, we can get a high quantity of this secondary metabolites. So the second process is the fed batch process. So if you see the fed batch process, actually this concept was brought by Yoshida et al. In 1973. The concept is simple. So consider a particular microbial cell is undergoing a substrate limitation in the second stationary phase. So we need a primary metabolite. So this we can get in the exponential phase. So to keep the cell in exponential phase, what we can do is to the batch culture, after a particular time interval, we can feed a small quantity or a certain amount of the fresh medium. So this will maintain in exponential phase, so we can get a product in a larger quantity. So this is the concept was actually lead to the fed batch process, but later on this process has been utilized in the penicillin production also. So if you see, if I need a benzyl penicillin, that is the penicillin G, at a particular stage, once the cell reaches, Penicillium chrysogenum reaches the stationary phase, the precursors can be added into the medias and feeding process, that is the fed batch process. So we can get the final product. So this is where the fed batch process started working in the fermentation industry. So the fed batch process is similar to that of a batch process. So initially the batch mode is established. Then based on the feeding strategies, the nutrients has been fed. So as you can see from the picture, if you see that the batch process is initially established, then the feeding is done from a feeder vessel with the help of a pump, which may be a peristaltic or a piston pump, we can use it. So if you see the feeding strategies, we can divide these feeding strategies into 4 ways. So one is the same dilution of the medium, which has been used for during the establishment of the batch culture is used, and it is pumped after a certain time. So what happens is, if I add a specific quantity of medium into the batch culture, once again what happens, the volume of the production process will increase. Other way, the major nutrient, that is the which substrate limitation is needed during the stationary process, that substrate only can be given at a particular volume inside it to the production vessel. This also can increase the volume. So the third way what we can do is, this limited substrate can be concentrated to a certain level that can be fed into the production vessel. This also gives a slight increase in the volume. But if we don't need any increase in the volume, what we have to do is we have to concentrate the limiting substrate and we have to add it. So this doesn't change a significant increase in the volume. So it will be insignificant in nature. So based on this, we can classify the fed batch process into 3 types. One is a variable volume, that is the strategy 1 and 2 will do. Second is intermediate volume, that is the strategy 3. And there is a fixed volume, that is a strategy 4. So what is the major advantage with this fed batch culture? If you see that the concentration of the limiting substrate can be maintained during the production level, so that the highest level of products can be achieved. So this somehow other ways will help us to have a control over the growth rate of the organism, so that the primary metabolite can be collected at a high rate. But still, there is need of another process, especially this primary metabolite is concerned, that is to maintain in exponential phase, we need another process. The fed batch process was not that much satisfactory enough. So the third fermentation process which has evolved based on this concept is that continuous culture process. So if you see the continuous culture process, so the objective of this process is to keep the cell culture in an exponential phase. So we have to prolong the exponential phase so we can get a large quantity of either the primary or the secondary metabolites. So how we can prolong this exponential phase is by adding a fresh medium. So if I add a fresh medium, the volume can be get increased. So to compensate that, we will withdraw the feed, that is fermented material. So we maintain the volume. This is how the continuous process works. So this continuous process can only be established if the growth is the substrate limiting, not the toxin limiting. So if it is toxic limited, nothing can be done. If it is substrate limited, we are supplying the fresh substrate. So if you see that this fermenter which is used for this continuous culture should little be modified from the batch culture, because same batch culture we can do the fed batch process, but for continuous process we need a modification. So the modification is done in such a manner that we have to feed a fresh nutrient, sterile fresh nutrient separately, and we have to withdraw the same quantity which has been fed inside, same quantity should be withdrawn from the fermenter. So this can be achieved by maintaining an overflow device. So we can keep a particular volume. So if it is overflow, that can only be sucked away. So this you can see from the picture in a continuous fermenter. So through pump 1, we are adding a feeding vessel is attached, so which supplies the nutrients. Once it reaches a particular level, the pump 2 gets activated, it will withdraw that fermented material. So what happens is the maintain of the volume of the culture will be there, so we can achieve the particular exponential phase. So the continuous culture process can be maintained by 3 ways. One is called as a chemostat, second is turbidostat, and third is a biostat. So if you see what is meant by a chemostat, so the term itself tells chemo means chemical, stat means static. So we are maintaining the exponential phase of the bacteria by using the chemical means. So the steady state condition of the microbial culture or a specific growth rate of the culture is controlled by diluting with the nutrient substrate. So we are supplying a nutrient rate at a specific volume so that the growth is maintained. So if you see very precisely what is happening is we are maintaining a continuous culture. So we know that the specific volume of the bacteria should be there. So we are adding a fresh media. So fresh nutrients is available for the growth. So because of this fresh medium input, we withdraw the fermented liquid. So the growth will be maintained. So the growth is maintained by the supply of the medium. So the medium is nothing but the chemical. So what we are doing is the growth rate of the bacteria is maintained through a chemical means. So we call it as chemostat. The second way we call as a turbidostat. So it is another way how we can maintain a continuous culture process. So the term turbidostat, turbidity static. If we take a fresh medium, it will be clear in nature. If I add a microorganism, when it grows, it produces turbidity. So what I am going to do with the help of this turbidostat is, in the continuous culture vessel, the turbidity will be maintained between a limit. So we add a fresh media, so the nutrient will be taken. If I am not adding a fresh media, the turbidity will start increasing. So to maintain the turbidity into a particular level, so what happens, we are adding fresh media and withdrawing the fermented media. So in other ways, what it actually tells is we are maintaining the concentration of the cell in the culture by using the flow of this medium within the narrow limits of the turbidity. And the final one is the biostat. So it is other way of turbidity, we are maintaining the cell volume. So with the help of certain biological indicators like the level of carbon dioxide or pH, we can maintain it. This is called as a biostat. So thank you very much for patiently listening about the different types of this fermentation process. So you continuously listen in my future lectures about the other interesting topics like the construction of fermenter, control, design, and various primary screening and secondary and the primary screening and secondary screening process also. So thank you very much.